eLTER, Integrated European Long-Term Ecosystem, Critical Zone, and Socio-Ecological Research.
Okay, so welcome to both Lars and Shangharsha to give the presentation on the site's spectral remote sensing. And Lars is a professor at Lund University. Shangharsha is a research engineer. They're both from the same department. Let me make sure I get it right. It's the Department of Physical Geography and Ecosystem Science at Lund University in Sweden. And so with that, I will hand it over to you, Lars and Shangharsha, to tell us a bit more about Site Spectral.
Okay, thank you. And thank you for the welcoming. I'll share my screen. So just a moment. Right, do you see a presentation now?
Yep.
Okay, so we are actually three people here from Site Spectral. It's myself, Shangharsha, and Jose, who is our new member of the group here. So you already mentioned that I'm from Lund University, or we are from Lund University, and that is actually my first slide, giving a little bit of a background to who we are. So it's a long name of a department, Physical Geography and Ecosystem Science, and that sort of reflects a little bit that we have a relatively broad spectrum of interests. So we are from the Earth Observation Group, but we also have other groups in our department dealing with different topics, focusing quite a lot on vegetation and ecosystems in general, mainly terrestrial vegetation, I would say, but also other issues related to physical geography and also human presence and how humans and climate alter the functioning of the ecosystems. So going over to the presentation itself, I will start by very quickly saying a few words about SITES. So SITES is the Swedish infrastructure for ecosystem science. It's of course not as large as eLTER, but it covers 9 stations, research stations, and they are distributed over Sweden. And you can see a little map of Sweden here, going from the south, where we have let's say temperate climate, up to the north through the boreal belt, up to the mountain range where we have tundra climate, basically. So it covers quite a nice range of climates. So SITES is coordinating and trying to, let's say, facilitate work within and between these stations, and there are many different activities going on. So here is the list of the stations, and you can see that they cover lots of different kinds of environment. There are purely agricultural stations, purely coniferous, but also more, let's say, general ecosystem stations that have a variety of different vegetation types and zones surrounding them. So more information you can get from the website of SITES. So SITES is about collecting data, and we collect open and quality control research data, and these are presented at the SITES data portal. And I will say a few words about the SITES data portal at the end of this presentation. But it also provides support to researchers. So it can provide field access. There are many of these stations have beds, so it's possible to visit the stations. There are laboratories, there is electricity, tools, instruments, and so on. And the staff, they can help with measurements and support to the projects. And then, of course, we try to maintain national and international contacts with different networks. So eLTER is very interesting for us, of course. There's also ICOS and Actris and some other networks that we are in contact with. So that is in general about SITES. And now I will focus in, no, sorry. I should also say that SITES has 3 overarching programs. And these are SITES Water, SITES Spectral, and SITES Aquanet. So SITES Water is specialized on water and hydrology measurements. Site Spectral, well, that's remote sensing and spectral measurements. And then Sites Aquanet is a more specialized, let's say, program for doing lake mesocosm experiments. So now I will focus in on Site Spectral. So within Site Spectral, we do data collection in different ways. So we have 4 main components, and one of them is near ground spectral measurements. The other one is mapping using drones. Then we have image collection using phenological cameras. And then finally, we collect satellite derived vegetation index data. And we have different aims. One of them is to provide data that facilitates ecosystem process understanding. And the other one is to provide reference data for remote sensing and modeling studies. And we try to present our data in a broad way so that it can be interesting for a number of different groups and let's say research specialities. So it could be useful for climate impact studies, biogeoscience, carbon and water flux studies, agricultural and forest science, field biology, ecology, for remote sensing specialists, and also hopefully for the general public. So in Site Spectral, we are producing a number of different data products. So you can see here the list of PhenoCam, multispectral sensors, UAV or drone, and satellite. So we, of course, generate these raw data. Some of them are images and some of them are time series data. But we also generate a number of different data products. So here we have products going from level 0, which is the raw data, up to more, let's say, processed and more advanced products. And the aim is that the higher products should be really readily available for and very useful for researchers who may not be experts in spectral sampling or remote sensing, but they should still be able to use these products. The other thing we are trying to do is to maintain data collection protocols, metadata. We do quality flagging of our data, and we try to build up a library of documentation so that everything should be clear and transparent to the users. So I will take you through these different data types, and I think it's difficult for me to answer questions during this presentation, so I guess we will reserve time for the questions at the end. So I will just keep on and present, and then you can save your questions for the end of the presentation. So first of all, we do multispectral and PAR measurements for long-term monitoring. So these are sensors that are relatively robust, and they are useful for outdoor usage, and we can leave them for long time periods. We measure from masts or towers down onto the ground, and we measure incoming radiation as well. These multispectral sensors, they measure in defined wavelength bands, usually red, near infrared, but also some other wavelengths that are useful for vegetation monitoring. We also sample photosynthetically active radiation, PAR, fluxes up and down, both above the forest and below the forest canopy. We also carry out sensor calibration of these spectral sensors, because we have noted that it's not very useful to keep these kind of sensors for a long time without any calibration. And we have opted to do the calibration ourselves. And this is for many practical reasons. It's usually relatively quickly done to do the calibration, and we don't have to send them off to some laboratory in the UK or somewhere for a long time period. So this goes quite quickly. And it's also a relatively precise method. So we have analyzed and compared different ways of doing calibration and generated a publication that shows exactly how this works. We have these spectral sensors mounted in different places, and here is an example from the very low density vegetation cover up in the Swedish mountain range near Abisko area. And you can see time series of NDVI that we generated from these data. And then we have different levels of products. So the sensors measure every 10 minutes, and then we aggregate these into daily products that are a little bit more easy to handle for users. So here are two examples of how one can use these data. And one of them is in improved process understanding. And in this example, I'm showing a little bit of GPP and radiation absorption features of vegetation. So we have a time scale here of the day of the year, so going from the beginning to the end of the year. And then we have temperature. This is again from Abisko area. And it's a relatively cold area, but a lot of light. And when the temperature is sufficient, the birch trees will start to grow, and they will generate, they will start to photosynthesize. And here is the curve of GPP, the gross primary productivity of the birch forest. So next to it or below it, we have some radiation measures that we have done. So we have the NDVI that tracks the GPP quite well during the growing season, but we also have some other interesting fluxes that can be useful. For example, the radiation absorption of the canopy, which tracks the NDVI and the GPP quite well, but also that of the understory vegetation. So this understory vegetation absorbs radiation sort of in when there is not so much absorption by the overstory. So there's this fluctuation between overstory and understory that is quite interesting to study. Right, the other example is how we can use this data for time series processing and to improve our methods for handling satellite information. So in this example, we have a relatively noisy time series of satellite data. And then we have the much smoother data series from the spectral sensor that we can use then to test out different ways of handling these rather noisy satellite data. Okay, so moving over to phenological cameras or PhenoCams. These are just RGB cameras, and I guess that many of your stations have such cameras, and they are quite useful for just getting a nice overview of the vegetation or whatever is going on at the site. So we can then define regions of interest and extract time series from this data. So again, we have different products. We have the raw imagery, and then we have quality filtered images, and we can then generate different kind of products, RGB images, green chromatic index, red chromatic index, and also for these regions of interest, we can generate time series data that sort of track the evolution of vegetation in time. We also have a quality flagging system with the data. So I'm just giving here the example of the phenological cameras, but for these we use the occurrence of snow, how many images are used for creating averages, the solar elevation angles. Those together form our flag value. So we give a flag value that goes from low to high data quality. And in this example, we have a time series of green chromatic index, and there are some noisy data here in the beginning and at the end of the time series. And if we fit a curve to these data, it will look very strange in the beginning and the end. But if we take these quality flags into consideration, we can do a much better time series fit to the data. So these are quite useful additions to these raw images, actually. Right, and then over to the next product. Here we are looking at images from UAVs, unmanned aerial vehicles or drones. And these are, of course, just images that can be useful. They are in different wavelength bands. So here we have blue, green, red, red-edged, and near infrared imagery. But again, we use these raw data. We start from those, and then we go on to generate a number of different products. So yeah, we have various types of user products that can be useful. We can do orthophotographs, we can do digital elevation models, we can measure vegetation height, and we can do image composites, doing combinations of different wavelengths. We can generate the NDVI and so on and so forth. So it depends a little bit on the usage, what can be applied. But we have looked a little bit at what could be the best practices and also how to handle data quality with these data. And we generated a number of studies that can actually be quite useful also for other users. But the other thing is that I think that some of these drone images can be quite useful for sort of presenting a field station in nice and popular ways to the general public or to researchers, providing an overview of the experiments or providing a view of the topography in the area or the vegetation distribution in the area. So it's not only this sort of more scientific use of these data, but also the more, let's say, popular use of the data that one can utilize. Right, so I'm moving on here with the next, or actually the last type of data that I wanted to talk about. And this is phenology and productivity data from satellite. So as you know, there are a number of satellites circling the Earth and also a large number of data products that are being generated. So we have the raw images, but also ready-made data products. And we have started out from one of those products that we think are quite useful for ecosystem researchers. And this product is one of the Copernicus land monitoring service. So it's a product that covers the whole of Europe. It's called the High Resolution Vegetation Phenology and Productivity Dataset, or HRVPP. If you're interested in this, you can simply Google for HRVPP and Copernicus, and you will find more information about this product. But anyway, the product is based on a vegetation index for green leaf area index called the PPI, or the Plant Phenology Index. And this is an index that basically tracks the abundance of green photosynthesizing vegetation in a canopy. And we track this in time, so we get sort of a seasonal curve like this. And then we can analyze this time series, we smooth it, and then we apply, then we derive different phenological metrics from these data. So some of these are the start of the growing season, the end of the growing season, the length of the season, the total productivity, so the area under the curve, the amplitude, so how high this curve is from the base level. We also look at the slopes and so on and so forth. So there are a number of different variables that are interesting and that describe the general, let's say, canopy development of an area. And these data now cover Europe at 10 meters spatial resolution, and they are available for 2017 to 2022. They can be downloaded freely, and we also do that for our project. So studies have shown that GPP can be estimated from the Plant Phenology Index. So we have recently conducted some studies that look at how GPP can be estimated for coniferous forest and deciduous forest and also wetlands using this simple index, the PPI. So there are a number of different models that can be used to transform satellite vegetation indices into GPP. And one of them is based on PPI. And actually these different methods for deriving GPP are not so, they don't give such different results. They are relatively functional, all of them. And this is a pretty simple way of doing it. So we feel quite confident that this PPI index is useful for tracking vegetation development and phenology, and that's why we have stuck with it so far. But we are also open to other indices. Anyway, here are some examples of phenological parameters at an agricultural site. So here we have picked out the start of season date. So it's the day of the start of the season. And the colors, of course, represent different starting dates. So we have the early dates in bluish colors and the late dates in reddish colors. The same for the end of the season day. Then we have the productivity, or the sum under the curve, as I was talking about, and the amplitude, which is the height of the curve. And these all then represent different sort of phenological parameters or stages for these agricultural fields. If we just look at the vegetation productivity for a number of years, we can see how it varied between 2017 and 2020. And as you probably all know, we had a very severe drought in Northern Europe in 2018. And we can see clearly from this index how in 2018 the fields were totally dry. And the average crop yield for Sweden was extremely low in that year. It was more or less a disaster for some of the farmers. So we have compared our productivity data that we get from satellite with those of official statistics for yield. And it turns out that they match pretty well. So again, we have a confirmation that these kind of vegetation data are useful for monitoring what goes on in vegetation. Right, so here is an example of what we do with these data. We sort of make them user ready. We cut out for our field stations an area that covers the field station, and we transform this or reproject it into the Swedish national reference system so that the users will be able to enter these data into their GIS systems. And then here are some examples of a couple of points where we have extracted the time series from these data. So they actually they peak at different times, reflecting that these are both spring growing crops and autumn growing crops in this area. Right. So we try to publish as much of this data as possible, and as soon as we have some quality checked data, and everything is published on the site's data portal. So here is the link, data.fieldsites.se, and here it's possible to sort of filter for different data types, and then these data can be previewed. They can be downloaded, and there will also be documentation together with the data. And we are gradually now filling up. So we started a couple of years ago, but still it takes time to develop all this quality checking and all the, let's say, securing that all the data are in good order before we upload them. So we are still sort of in the process of filling with more and more data. So now I thought that I would leave over to Shangharsha, my colleague, to say a few words about how he has applied these data. So I will do the clicking here, and you can just, Shangharsha, you can tell me when you want me to change to the next slide.
Sure. Thank you, Lars. So by this time, you now know a little bit about site Spectral, what sort of data we have been collecting, and what type of data products we have been producing and making it available for the researchers. And now here I will give few examples of how the data looks like and how the data can be used to do some science. This case study will be showing the PhenoCam data over mountain birch forest that we have in northern part of Sweden at Abisko Research Station. as we all know, the phenology of vegetation, they are dependent on various environmental factors, such as temperature, rainfall, as well as the amount of light that is available for the vegetation. And with this kind of data sets, we can also not only monitor phenology and the vegetation dynamics, but also we can see how the vegetation reacts under extreme events, such as infestation by defoliating insects. In this picture, the left picture shows the picture from the springtime, when we can see the greenness in the picture. And on the right side, we can see one of the pictures captured by PhenoCamera during fall, where we can see the vegetation looks red. So by looking at the time series of these pictures that are being collected by the PhenoCamera over a year, we can actually compute the amount of greenness or the amount of redness that is available within the camera. Next slide, Lars. So here we process these every half an hour temporal resolution PhenoCam images, and then come up with the different vegetation indices, the green chromatic coordinate, which provides information about the amount of brightness in green channel and how it varies throughout the year. And also we have temperature data from ICOS sensors located at Abisko as vertical bar. So in the graph, we can see the time series of green chromatic coordinate from 2012 until 2021. This shows that we have a very long time series data available for PhenoCam sensor. So if we look at this plot, we can see something is happening in the year 2012 and 2013. Of course, there are some changes in other years as well, but the one in the year 2012 and 2013, when there was insect attack for this birch forest, and we can see slightly different time series of green chromatic coordinate. Next slide. And the same, this is the time series of red chromatic coordinate. This gives us an idea of how the redness differs over the year. And the peak in most of the curve, you can see one peak around September or October. And this peak shows the time when the leaves in the vegetation are completely red. And this marks the end of season for the birch forest. So I will go a bit more by demonstrating how these time series datasets can be combined with temperature and come up with some interesting signs. Next slide. Yes. So we try to derive seasonality metrics from those time series data that we have produced by using time series of PhenoCam pictures. And then this start of season date information, we try to correlate with temperature and amount of radiation that is available for the vegetation to do the photosynthetic activity. So we try to compute cumulative negative temperature at negative temperature at the start of season date. So we have data from 2012 until 2021. We have a start of season date computed for each of these years, and then we try to find out the relationship with cumulative negative temperature. And we can see from this graph that there is strong positive relationship with start of season date and cumulative negative temperature. Similarly, next. So the end of season date, by looking at this scatter plot, we can see that the seasonality event such as end of season is often triggered by light. As you can see in the plot, the amount of cumulative PAR seems to have a strong positive relationship with when the season ends. Next slide. And here, interesting to see that the start of season date for the birch forest is also quite well related with amount of light that is available during the start of season. And in all these plots, we can see the year 2018, which is the drought year in Sweden, as Lars mentioned, and we can see that that it looks a bit different compared to other normal years. So we have earlier start of season and also earlier end of season compared to other years. Next slide. So that was one case studies, and then now I move into next case studies that we carried out. So here I will present how this multi-scale data sets being collected from different sensors can be used to compare forest phenology. On the right side, you can see a small diagram where you can see different sensors, for example, satellite, multispectral sensors. We also have PhenoCams as well as the multispectral sensors. So the PhenoCams and multispectral sensors, as Lars already discussed about, collects data at very high temporal and spatial resolution. One of the good aspects of this kind of having this kind of data at various scales is that we will have the opportunity to look at common overlapping region and derive vegetation metrics, also seasonality events for this overlapping region, and try to see if there is any similarity in between or if there is any difference. So when we derive vegetation indices, such as the commonly used NDVI from different sensors, not only NDVI, but there are also some other vegetation indices, which is based on just the visible bands like red, green, and blue. Plus, if you click once, there will be another graph. Yes. So looking at these vegetation indices derived from near surface sensors like these PhenoCam, UAV, multispectral sensors, and if we compare them with satellite sensors, we see very good correlation between the vegetation metrics derived from these sensors. Next plus once. Yep. So yes. And the data derived from the sensors, they are sort of complementarity to each other. And this somehow show the potential to upscale vegetation monitoring, vegetation dynamics, as well as vegetation seasonality from near surface sensors to upscale it up to the landscape scale by using combination of these proximal sensors as well as satellite sensors. So the details can be studied in the link that is presented here. I think this is the last slide. Yeah, thank you all for your attention.
Okay, so yeah, that was the last one. And I think one thing that I would like to stress here is that we are trying to make this data available in sort of user-friendly format, so that one should not have to be a remote sensing specialist to be able to use remote sensing data, but we try to produce products that are quite easy to manage and to use. So that's it for us. Thank you very much for your attention.